The first example of biotechnology that I want to talk about is what we call molecular cloning. And so cloning is making an exact copy. So making an exact copy of a protein. And the way that this has been done is by sticking genes into bacteria. So we use the bacteria, and bacteria are interesting because instead of chromosomes, they have plasmids. So they don't have chromosomes, they have plasmids, and these are circular pieces of DNA. So they have their own genetic material, but wouldn't it be nice to stick a human gene into their genome and have bacteria produce a human protein, right? So a human gene is inserted okay? And then the bacteria produces human protein. Does anybody know what human protein was produced first? It's a hormone, and it's used to treat diabetes. Insulin. Okay. So the first way, you know, it used to be that they would have to take, I think they used pigs, and they would try to extract the pancreas from a pig, and they would try to get the insulin out of it. But you can imagine that pig insulin might be a little bit different than human insulin. And then, you know, it's kind of a tedious process. So if you can get bacteria, if you could just like brew, you know, when we think about fermentation, like cheese making or alcohol making, what we're essentially doing is we're growing bacteria, right? And so if we can just grow bacteria and then have them produce the insulin, then we can get the insulin from them. So human insulin. So in your book, they have an image of this, right? So this is the circular DNA. This would be the DNA of a human, for example. And we're going to talk about how we cut the DNA into particular sections. So we use molecular scissors. So we use enzymes that can cut the DNA. And then that foreign DNA gets inserted into the plasmids of the bacteria. And the bacteria reproduce, and they pass that foreign DNA on to their offspring. Bacteria reproduce asexually. So they just divide asexually. They're really interesting. They can, instead of sexual reproduction, what they do is that they can actually pick up DNA from their environment. So if they don't need a gene, they can actually just get rid of it, and then they can pick it back up in their environment. So that's how they get new variation. But anyway, so the bacteria just clone themselves, right? So we can see that we can find the bacteria when we grow them on an agar plate that have the foreign DNA. And then we can use those bacteria to produce human insulin. So that is molecular cloning. Okay. Therapeutic cloning is used to obtain stem cells. So we all have stem cells in our body. Stem cells are unspecialized cells that can become anything. So like we have stem cells, they even discovered that we have stem cells in our fat. And they discovered that by they looked at people's fat that had been liposuctioned out of them, and they found that there was stem cells that could repair and replace any type of cell in the body. So you could actually get stem cells. We get stem cells, we can get them from embryos. We can also get them from the umbilical cord. So they can be found in embryos and the umbilical cord. So there is an option when you have a baby. They say, do you want some of the stem cells from your umbilical cord frozen so that when your baby grows up and might get sick, so let's say maybe they have a virus that attacks their pancreas, or maybe they have neurological problems, maybe we could insert those stem cells back into your child. But it's very expensive. So you can send your umbilical cord away, but you have to pay them money to keep those cells frozen, right? And then you might actually never use them. Okay. So they can also be found in most tissues.
If you can find it in fat cells, why would you need the umbilical cord?
That's a good question. Maybe they are going to phase that out. It's probably easier than to have fat suctioned out and to find to sort through all the cells to find stem cells. That's because umbilical cords can have a lot of stem cells in it. So maybe that's the reason, I don't know. Okay, so can we create stem cells is the question. So creation of stem cells that are unique to you. Okay, so that have your genome, right? So let's say your parents didn't pay to have your umbilical cord, and you don't want to go in and have tissues removed to find stem cells. So there is the option of creating stem cells and then using those stem cells therapeutically to treat illness, okay? So we take a body cell and we remove the nucleus. We then insert the nucleus into an egg that is enucleated. I don't know why they put an E in there instead of an A. It's anucleated, right? But I don't know why they put an E in there. So enucleated means the egg has had its nucleus removed, because that nucleus would be haploid, right? So if I can take one of my body cells, so let's say they take a sample of my skin cells deep down, or maybe blood cell might be better, a white blood cell, for example, and they remove the nucleus, and then they put that nucleus into an egg, and it could be my egg, or it could be somebody else's egg. And then so if you're male, you can still do this, and then they stimulate it, generally with an electric shock. So they provide an electric shock, and that stimulates mitosis. Okay, so then you produce a multicellular embryo that looks like this. There could be more cells than this, but this is early on, right? And so you could theoretically then harvest these embryos, take out the stem cell, and then put the stem cell back into my body, right? So the stem cells are used therapeutically to treat disease. Okay, so you can imagine that this is pretty controversial, right? Because some people believe that even early embryos are living creatures, right? And that it would be unethical to harvest, to create embryos just for the sake of harvesting stem cells from them. Okay, this is actually the same mechanism that we would use to do reproductive cloning. So instead of using it to treat disease, let's say we took this embryo, right, and we put it into a female's reproductive tract. So we put it into the uterus, right? And then we would have a clone. And this is an example of a reproductive clone. So that would be the difference between therapeutic cloning and reproductive cloning is that the embryo isn't developed into a fetus, isn't born, and is not a clone of you. Now I have to mention that this is a very, this ability to do this is not easy. And so it's not like you could do this really easily, like we thought it was going to be really easy to do when we first did it. We thought everybody was going to be cloning and we were going to make regulations, but it is very inefficient. So you have to do a lot of these to actually get specifically to get an embryo that would actually be able to be born and to possibly reproduce. Okay, so it is inefficient, right? The mechanism, it takes a lot. Okay. The other thing I wanted to mention is that if we look at these embryos, there's been a lot of controversy over the use of those embryos and the destruction of them. Because when you look at fertility clinics, they produce more embryos than they could possibly use, right? So when we go to a fertility clinic and they take the eggs out of the female, they give you a drug that causes you to ovulate. It's a super ovulatory drug. So you might ovulate like 8 eggs at one time, right? I think it probably would be painful. And then the eggs and the sperm come together in a test tube, right? And then they produce all these embryos, and they just take one embryo and they put it into the female that wants to get pregnant, okay? So then they have these leftover embryos. And so the idea is, what do we do with those leftover embryos? And it was against the law to use them medically, so to study them and to use them in experiments was illegal until recently. And I'm not sure if Trump has made a decision about the use of them, but this would be a source of stem cells for research, right? If you went to the fertility clinics and you were able to obtain those extra embryos and they weren't destroyed, then you would be able to use them in your research. So does anybody know what was the first organism and what was their name that was successfully cloned reproductively? Dolly the sheep. Does anybody know why she was called Dolly? Anybody know? It is. And so why is it after Dolly Parton? Hmm? Big hair? Oh, I didn't know about that. So Dolly is actually the cell that they took it from was a mammary gland cell. So it's a little bit more risque than that. Hair, it's the big boobs. Okay, was used to clone, right? So that mammary gland cell, the nucleus of the mammary gland cell was removed and put into the enucleated egg cell, shocked, right? And then you got Dolly the sheep. So one interesting thing about Dolly the sheep is that she did not live very long. So she seemed to have accelerated aging and died at 6 years of age. And they can live to what, 10 maybe? So she died young for relatively to the other sheep. So can anybody think of a mechanism which would cause her to have accelerated aging? So there could be a higher mutation rate, but that's not it. They stop dividing. And why will they stop divide? Shortened telomeres, right? So the mammary gland was, the cell was taken from an adult, right? And so there was shortened telomeres. Right? So this meant that there was a short mitotic clock, right? So this is related to the mitotic clock. Now, interestingly, they did breed Dolly before she died, and all of her progeny had normal telomeres. So in her body, somehow in the process of developing eggs, she added the telomeres back on, and we all know that that happens. So we know that that happens in the gonads. We know that happens in the ovaries and the testes, is that the telomeres get added back on, so that the cells that are passed on to the next generation have the right equivalent, right? Okay. So our offspring were okay. So in your book, there is a diagram that shows this process, right? So they used two different types of sheep so that they could keep track. They wanted to make sure that it wasn't the surrogate mother that had a baby, right? We wanted to make sure that the nuclear donor, which supplied the mammary gland cells, right? The nucleus was added to the egg. They give it a shock, right? They produce an embryo. They put the embryo into the surrogate, and then Dolly was born. And she was of the same breed as the nucleus donor, the nuclear donor. So one reason why you might want to do this is that if you have a particularly robust individual, perhaps you want to maintain its genetic robustness, if we're talking about breeds of animals, right? So you might want to make a clone of it. But another reason we might want to clone these organisms is if they are genetically engineered. And if we have a genetically engineered special organism and then we clone that organism, then it passes on its being genetically engineered, right? So we're going to talk about that. So I just looked it up, and they think that this has not become really popular. They thought it was going to be a really popular thing. It's a lot harder and it takes a lot more time and effort than it's worth in most cases. So there's like maybe 1,000 cloned individuals of all different types of animals around at any given period of time, like in the United States, right? So it's not a very popular method. And we do not think humans have been successfully cloned. So I'm going to skip over DNA fingerprinting.